let us talk about another important phenomena which is called catabolite repression. So the function of beta galactosidase in lactose metabolism is to hydrolyze lactose, and the hydrolysis of lactose yields its two monomers which is glucose and galactose. If the growth medium contains both glucose and lactose, then in the interest of efficiency there is no need for a cell to turn on the lac operon, because glucose and lactose are present, so the lac operon system will not be turned on. Monod in 1940s showed that cells behave according to this logic. E. coli cells incubated in the presence of glucose and lactose do not start to make beta galactosidase until all the exogenous glucose is consumed. So if glucose is present, then why a cell will have a need to break lactose to yield glucose? These findings were later extended to lactose permease and transacetylase, the two other proteins which are specified by the lac operon. The reason that lactose enzymes are not made when glucose is present is that no lac mRNA is made. So if no lac mRNA synthesis, ultimately there will be no production of the proteins specified by this operon. Transcriptional level inhibition of the lactose enzymes and a variety of other inducible enzymes by glucose or any other readily available carbon source is called catabolite repression. So अगर मीडियम में जहां सेल्स ग्रो कर रहे हैं वहां पे ग्लूकोज या कोई और रेडिली अवेलेबल कार्बन सोर्स जो है, कोई और रेडिली अवेलेबल शुगर जो है मौजूद है, तो इस सूरत में ट्रांसक्रिप्शन लेवल पे इनहिबिशन हो जाएगी, जैसे कि लैक सिस्टम की इनहिबिशन हो जाती है. तो इस फिनोमिना को कहते हैं कैटाबोलाइट रिप्रेशन. The mechanism by which glucose inhibits beta galactosidase synthesis remained a complete mystery for about 20 years after Monod first observed the phenomena. Richard S. McMahon and Earl W. Sutherland found an important clue to the mystery in 1965 when they observed that the intracellular concentration of 3,5-cyclic adenylate or CAMP drops from about 10 to power minus 4 to 10 to power minus 7 when glucose is added to a growing culture of E. coli. So जब इकोलाई ग्रो कर रहा है तो उसमें जब ग्लूकोज बाद में ऐड किया जाए तो उसका CAMP लेवल जो है वो ड्रॉप हो जाता है. जेनेटिक स्टडीज कंफर्म द इन्वॉल्वमेंट ऑफ CAMP इन द फिनोमिना ऑफ कैटाबोलाइट रिप्रेशन. 2 mutant classes were isolated that couldn't synthesize lac enzymes when cultured in medium containing lactose but not glucose. So in this case lactose is added in the medium, so they must turn on their lac system, but these mutants were unable. Class 1 mutants regained the ability to synthesize lac enzymes when CAMP was added to the growth medium, but class 2 mutants didn't. So क्लास 1 म्यूटेंट भी ऐसा म्यूटेंट था जो कि लैक सिस्टम को ऑन नहीं कर सकता था, लेकिन अगर CAMP उसमें ऐड किया जाए कल्चर मीडियम में तो इट वाज एबल टू सिंथेसाइज लैक एंजाइम्स. Subsequent studies showed that class 1 mutant have defects in adenylate cyclase, which is an enzyme that converts ATP to CAMP. Adenylate cyclase exists in an active form, that is phosphorylated, and an inactive form, that is dephosphorylated. So ये जो adenylate cyclase है इसकी दो फॉर्म्स हैं, एक्टिवेटेड और इनएक्टिवेटेड. तो एक्टिवेटेड फॉर्म जो होगी वो फॉस्फोरिलेटेड होगी और इनएक्टिवेटेड फॉर्म जो है वो डीफॉस्फोरिलेटेड फॉर्म होगी. यहां आप देख सकते हैं कि एटीपी जो है एटीपी इज कन्वर्टेड टू CAMP with the help of this adenylyl cyclase enzyme. So in this way CAMP synthesis from ATP takes place. Class 2 mutants have defects in a protein that binds CAMP. This protein called the CAMP receptor protein or CRP, or it is also called catabolite activator protein or CAP. It is encoded by the CRP gene. In vitro studies have shown that CRP and CAMP, they combine and they form a CAMP-CRP complex, and this complex is needed to activate the LAC system.